How's everybody doing? You're doing better than I am, huh? Yeah. Okay, so we are moving our way through protein structure, and one of the things I wanted to say something about was there's a couple things about amino acids I didn't finish saying something about, so I'd like to make sure that you're aware of them. And there's some terms that I use, and they involve some calculations as well. So one is the term PI. You probably have seen it on some of the problem sets, and I haven't explained that to you. So I want to say a little bit about what that is. So PI is a pH. It's a special pH. It's a pH at which a molecule, that's any molecule, has a charge of exactly 0. The PI for a molecule must be calculated precisely. It's not something that some of the estimates that I give you will work. Okay? Now, one of the estimates that I've given in the notes, I haven't talked about it specifically in class, but I'll mention it here also, is there's an estimate you can make for a charge of a molecule based on its pH and its pKa. And this estimate that I'm going to give you for the charge of a molecule only works for estimating. It does not work for calculating PI. Okay? So here's the estimate. This estimate is one I will not give you on an exam. You'll need to know what I'm getting ready to tell you. I give you the formulas, I give you various things, but I'm not going to give you what I'm getting ready to tell you. And that is that to estimate the charge of a molecule, if the pH is 1 or more units above the pKa of the molecule, or of a given group, if we have a molecule that has more than one, if the pH is more than 1 unit above the pKa of the group, then the proton is off. And if the pH is 1 or more units below the pKa, then the proton is on. And that's good for estimating charge. And we'll use that frequently. So you're going to estimate charge of amino acids. You may estimate charge of proteins and so forth. That's very good for that estimate. It's not good for calculating PI. Now, I'm not going to go through how you calculate PI here because I have a video online that shows you how to calculate PI. People think that that the only videos in the class are the ones that are under this column on the left. In fact, if you look on the right, you'll see problem-solving videos here, and there's more as you go further down. Practice protein charge problems here. If you watch those videos and you work through those problems, you will have some good practice. So how do you calculate the PI? The PI is calculated as the average of the two pKa values around where the molecule has a charge of approximately 0. Okay? So when you look at a titration plot, you'll see a place where a molecule has a charge of approximately 0. It's the average of the two pKa values around that. That means that you can't just add up the pKa's and average them. You've got to do a plot and figure out where that approximate 0 point is and then average the two pKa values on either side of that. If you watch the video, it should help you. And if you have questions after that, then of course come see me. All right. So PI is a very important thing because it's the place where a molecule has a charge of exactly 0. And we'll see later that there's some times when it's useful for us to know when a molecule, a protein, an amino acid, or whatever, has a charge of approximately 0. We'll see when that occurs. Okay. Another term I haven't used that I've mentioned also, I think, in my highlights, but I'll bring it up here, is the term zwitterion. Z-W-I-T-T-E-R-I-O-N. You can bet if I spell it for you that its spelling must be important. Okay? Zwitterion is a molecule whose charge is 0. Right? So you might say, well, it's the same thing as PI. No, PI is a pH at which a molecule has a charge of 0. A zwitterion is a molecule whose charge is 0. Okay, two different things. Okay, excuse me. All right, so I want to make sure I got those out there because there are some problems that I've got, practice problems to work, and who knows, you might even see something like that on an exam. So it's entirely possible that you'll have that happen. So I want to make sure that you're familiar with those things. Questions about that? No questions? Okay. Question? Yeah? Same thing. question was, do I mean zero charge or net zero charge? And the answer is same thing. A molecule has a zero charge if its net charge is 0. Yep. Okay. All right. Well, let's see. So last time I got to the point of talking about Ramachandran plots, and I don't want to make too big of a deal out of Ramachandran plots, all right? Because students go, oh my God, what do I have to know about this, et cetera, et cetera. Well, what you have to know about Ramachandran plots is, first of all, they're theoretical plots that tell us something about the structure that a protein can have. Okay? It tells us something about the structure. All right? So this was the Ramachandran plot that I showed you last time, and I noted that there was an awful lot of space on a Ramachandran plot where structures are not stable. Okay? Now, I'll remind you that a Ramachandran plot has a plot of phi and psi, and there's 360 degrees for each one, from plus 180 to minus 180, from minus 180 to plus 180. These are rotational angles of the two bonds around the alpha carbon. I don't care if you memorize which one is which. That's not an important thing. But what is important is the fact that there's only certain rotations around those bonds that give rise to stable protein structures. Anything that's colored up here is relatively stable. The darker it is, the stabler it is. All right? And the whites are basically regions of rotation around those bonds that are not stable. Okay? Now we're going to see in just a little bit that some of those rotations correspond to very specific structures that are commonly found inside of proteins. Commonly found inside of proteins. Well, that's kind of nice to know, that a theoretical plot matches up with what proteins actually have. And that not only that, but we see commonality. We see these structures over and over and over inside of proteins. And we're going to talk something about that. So that's the sort of gist of a Ramachandran plot. You're not going to draw a Ramachandran plot. Okay? You're not going to interpret a Ramachandran plot. But you should know something about what a Ramachandran plot tells you. Okay. Well, I've just finished talking about what I've been discussing so far, which is primary structure. I'll remind you that primary structure was the sequence of amino acids within a protein. And I said that the sequence of amino acids in a protein determined everything, ultimately, about the protein. Okay? Well, we're going to turn our attention now from primary structure to secondary structure. And as we go higher levels of structure, we start thinking about interactions between amino acids that are further and further away. Primary structure was the sequence, and the sequence related to the immediately adjacent amino acid. This amino acid, this amino acid, this amino acid in a protein. Okay? There's no distance. It's immediate. The very next one is the one that matters, right? Well, with secondary structure, we expand that view out to about 10 amino acids. Okay? So secondary structure, I'm going to give you a definition here. Secondary structure is a regular, repeating structure that arises between amino acids less than 10 apart. Less than 10 amino acids apart. It's a regular, repeating structure that arises as a result of interactions between amino acids that are 10 or fewer apart. Now, that's a definition, all right? In reality, what does secondary structure mean? All right? Well, for our purposes, we're going to talk about two primary types of secondary structure, and we're going to see that they line up with that Ramachandran plot pretty well. The two primary types are known as alpha helices and beta strands. And these were the structures that Linus Pauling discovered and received the Nobel Prize for. It was his first Nobel Prize. The structure of a protein. Okay? Well, here are 4 depictions of an alpha helix. An alpha helix, as its name suggests, is a helix. And you see 4 different ways of portraying that helix that protein chemists frequently draw. This one I kind of like. This is called a ribbon structure. It's sort of a combination ribbon and atom structure. If it didn't have those atoms on there, we would just call it a ribbon structure. But ribbon structures are really good for giving us the shape that a structure would have. If we wanted to be precise, we would actually look at the second structure here, and this is the depiction of what that short section of peptide would look like if we looked only at the atoms. It's a little harder to look at, but it also has an advantage that in this structure we can see some of the forces that are stabilizing this alpha helix. The forces that stabilize an alpha helix are hydrogen bonds. Hydrogen bonds stabilize an alpha helix, and you can see them in this structure here as little green dashes. These little green dashes arise between amino acids that are about 4 to 7 amino acids away. That's less than 10. Notice that the definition I gave you said it's a regular repeating structure. What does that mean? Well, a coil is a regular repeating structure. That's what this alpha helix has. That's what this is trying to portray. What else can we say about an alpha helix? Well, what we can say also shows up nicely in both C and D. If we look at those green dots that are on there, on each of these, the greens correspond to the R groups. Now, if we look at this, we see in an alpha helical structure that the R groups are pointed outwards from the helix. That's nice. Some of you have asked me individually, and I think somebody asked in class also, do some amino acid side chains affect the structure of the protein? The answer is yes, they do. We try to put 2 bulky side chains next to each other that it may very well disrupt and prevent an alpha helix from forming. Now, we can actually use computer programs to analyze an amino acid sequence and predict with a reasonable amount of certainty if a given segment of a protein is present in an alpha helix. A computer program can do that using the knowledge that we have of the R groups and so forth. Okay? So that's something that a computer can do for us quite readily. This shows some of the interactions between the hydrogens and oxygens and hydrogens within an alpha helix. And when we twist these around in 3-dimensional space, something that looks like it's far away in primary sequence actually ends up being pretty close to each other in secondary structure. Okay? So this and this are fairly close when the alpha helix forms. Okay. If we look at the Ramachandran plot, we see, there are 2 places on the Ramachandran plot where the helices can form. One is a right-handed helix, which is very common, and the other is a left-handed helix, which is very rare. But they appear right square in the middle of predicted regions of stability by Ramachandran. Okay. This is a nice depiction of a protein that's full of alpha helices. Okay? Now you'll notice that this alpha helix, each alpha helix doesn't just go forever. It'll have a little bend sometimes. Sometimes it'll have a bigger bend. And we'll talk about those bends when we talk about tertiary structure. But suffice it to say that there are proteins that have almost exclusively alpha helical structure. Not all proteins, but some proteins have that. All right, another common secondary structure, and the second one described by Linus Pauling, is known as what some people call beta sheets. I like to call them beta strands. If we take beta strands and we arrange them together, we get a sheet. So a sheet is an arrangement of a bunch of beta strands. Okay? We'll start here with the Ramachandran and look, oh, wow. The beta strands structure right square in the middle of the theoretical region of stability in the Ramachandran plot. What is a beta strand? Well, let's talk about that. Here's what a beta strand looks like. It's a little hard to tell from this figure what it looks like, but a beta strand is like a helix in 2 dimensions. A helix goes round and round and round. A beta strand is like, it's also called pleats. Up, down, up, down, up, down, up, down, up, down, like that. Okay? So it's a flattened 2-dimensional version of an alpha helix. All right. This depicts that up, down pretty well. Okay? Up, down, up, down, up, down, up, down, up, down. Okay? You see that the greens, again, are the R groups. And you remember from the structures that we saw before of the R groups that they were on opposite sides every other one. So here's one in the back, front, and here's the central part with the carbonyl group, the alpha carbon, and the carboxyl group, up, down, up, down, up, down, up, down, up, down. You can see like that. Now, what this shows is interactions between 2 different beta strands. 2 different beta strands interact like this. They're going to give rise to a beta sheet, which is a set of them. Now, when we look at this, we see that what stabilizes the beta sheet are hydrogen bonds again. Hydrogen bonds are holding together beta sheets. We might have a whole stack of these. We might have 10 of them. If we had 10 of them stacked up like that, or maybe even more, we would have something known as silk. Silk is full of beta sheets. Okay. Parallel versus antiparallel simply refers to whether the carboxyl and the amine are like this, or if the carboxyl and the amine ends are like this. These are parallel, these are antiparallel. If this is carboxyl end, this is amino end, this is carboxyl end, this is amino end, antiparallel, parallel. That's all it means. Okay. The overall structure, it doesn't matter if they're parallel or antiparallel. Here is an enzyme that has a structure that has beta sheets arranged rather nicely into what we call a barrel. Okay? You can see that this barrel and the blue parts are the strands that are all oriented with respect to each other. They create a barrel, and this barrel on the inside is fairly protected from water. Barrels like this we occasionally see when enzymes need to have water or protection from water for something, or they need to have a reaction contained within a certain space, and barrels work very well for those things. Okay. Now, we've talked about, I showed you the alpha helix, and I said, well, alpha helix will go for a ways, and then we see a bend, or we see something else that sort of interrupts that. So I want to say a little bit about things that interrupt structures, specifically things that interrupt secondary structures. All right? Now, I've told you that there are some amino acids that really can cause some changes. One was, I said, things that have very bulky R groups. If you put 2 of them together, they may not fit very well, and that may cause a bend if we had an alpha helix going on, for example, or a beta strand. All right? Other things that I've said can disrupt protein structure is proline, because proline, you may remember, was the amino acid that had the R group that came back around and attached to the alpha amino group. And I said it was less flexible. Well, what you see on the screen is something that arises very commonly as a result of the appearance of proline within a polypeptide sequence. I here refers to one amino acid. I plus 1 is the next amino acid. I plus 2 is the next. I plus 3 is the next amino acid. So here's a series of 4 amino acids. And what you see on the screen is a depiction of a bend. Okay? Instead of going off in a helical nature or on a pleated nature, this bend is happening right here. And the bend is happening because something is not consistent with that regular structure that we saw for an alpha helix or something that we saw for a beta strand. As I said, most commonly bends will arise in a structure like this because of proline. Proline is not flexible. And also commonly, we will see in a structure like this glycine. Glycine, you may remember, was the amino acid that had the smallest R group. And it's glycine that allows for the most flexibility, the most options that are there. And glycine may oftentimes also be in a structure like this. Now, bends are important for us to consider when we start considering the next level of protein structure, and that's tertiary structure. So I'm leading up to getting into tertiary structure. I'm not going to talk about tertiary structure at the moment. I'm going to save that for a few minutes from now. But suffice it to say that tertiary structure really has a lot of bends in it. Yeah, question?
What would make a protein an alpha helix and what would make it a beta sheet? What depicts that?
Yeah. What depicts it or what amino acids would favor it?
Yeah, what amino acids?
Okay. So it's a good question. So what will tend to favor an alpha helix versus a beta strand? I think I've got a link on here. I will show you that someone's compiled a list of all the different amino acids and their tendency to be in a structure that's alpha helical or beta strand. And so there's no absolute rule, first of all. So there's no rule that says tryptophan can't be in an alpha helix, okay? Because tryptophan can be in an alpha helix. Its likelihood may be low depending upon the amino acids surrounding it. And when we start thinking about amino acids surrounding it, it's what makes our predictive abilities a little bit harder to do. So if I could say, for example, tryptophan is not in an alpha helix, you would say, okay, well, I'll mark that one off. But it can be, okay? So, but I'll show you a table that shows a preference for one versus the other, and then you'll get a bit of an idea of that. Is that okay? Okay. Yeah, another question. Do you ever see a hydrogen bonding occurring with something other than something in the amine group? Let me think about that for a second. So you will not get it within the backbone because in the backbone, the only things that can hydrogen bond there are those that are linked to the amine because you have to have a partial positive charge. And there's nothing in the backbone that would have a partial positive charge except for that. If we think about R groups, yes, we can have that because R groups can have a variety of things that are inside of them, but not inside the backbone. Okay? Good question. Okay. I've already talked about that. Okay. All right. Now, I didn't want to talk about tertiary structure yet because I wanted to talk about a special class of proteins that are interesting because they only have primary and secondary structure. Most proteins have at least primary, secondary, and tertiary. Some will have quaternary, some won't, we'll see. Most proteins have primary, secondary, tertiary, but there's a group that has almost exclusively primary and secondary and very, very little, if any, tertiary. This group of proteins that has only primary and secondary structure, known as fibrous proteins. And as their name would suggest, they're fibers. So we could imagine something that has an alpha helix and it just keeps going and 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 going, and your hair would be a really good example. Okay? So your hair is full of alpha helices that just go on and on and on and on and on and on. Now, an individual hair is comprised of course, many individual alpha helices all meshed up and intertwined and so on and so forth, so it's a little complicated. But the important point is that they don't fold. They don't have much in the way of tertiary structure. If you have curly hair, you may have some slight bends that happen as a result of some disulfide bonds that happen that cause some bending to happen within that overall bigger structure. All right? So people who go, and people used to go do this and get permanents and things like that to get their hair styled and curled and so on and so forth, would come out of the hairdresser smelling like stinky because the hairdresser had to break those disulfide bonds. Those are sulfur bonds. They had to use sulfur compounds to do that, and that's why your hair stinks after a permanent. Those bonds aren't permanent, though, as you discover if you have one of those, so they will come apart, and they will go back to its original shape. So even though you get your hair curled, it doesn't stay curled forever. If you have curly hair, your hair has already assumed those structures in its most native form, and it's hard to change that. But again, that's what a permanent does. Okay, now let's excuse me. Let's look at some of these arrangements. So these are coils of coils. Here's an AlphaHelix, and here's an AlphaHelix coil with another one. We can imagine that hair might have many of these guys, many of these bundles that come together to ultimately make a hair that we can see. Okay. I'll talk about that another time. Let's talk about, oh, don't do that to me. Okay. One of the most interesting of the fibrous proteins is the most abundant protein in your body. Okay? One of the most interesting of the fibrous proteins is the most abundant protein in your body. And the most abundant protein in your body is known as collagen. Collagen is a very odd fibrous protein. I'll tell you why in a second. But it is a fibrous protein. It has very, very little tertiary structure. And it, too, is a coil of coils. It's three coils coiled together. Okay? So that was actually, here it is. That's one depiction of it. We see three coils coiled together. We see one strand in blue, one strand in red, and one strand in yellow. Okay? Collagen is the glue that holds us together. It attaches cells to each other. It attaches bones to muscle and a variety of things. Okay? So collagen is really an important glue for us. And that's why we have so much of it. Very, very important for us to have that glue that holds us together. Okay? Now, I'm going to tell you something that's kind of cool and has medical implications about collagen. All right? Collagen, you saw, was in fact got the wrong one again, was a coiled structure. And we can imagine that we might have one coiled structure now that bundles with another coiled structure, and we get, sort of super order of these coiled structures. It turns out that there is a reaction that occurs to modify some of the amino acids in collagen. We haven't talked about modifying amino acids. It's appropriate now we talk about that. Some amino acids in proteins get modified after they're put into a protein. They're modified after they get put into a protein. In collagen, the two most common are lysine and proline. After they're made into the collagen structure, they're modified by putting a hydroxyl group onto them. Modified by putting a hydroxyl group onto them. That reaction requires vitamin C. One of the reasons you have to have vitamin C in your body is so that you can make collagen that has those modified prolines and lysine residues there. I'm going to tell you why they're important in a second, but you need to understand what they are first. Okay? Now, you're probably sitting there before, as I'm saying this, scratching your head going, but Kevin, you said that proline disrupted. I'll tell you what's disrupting right here is the collagen. Why do I keep going back to that one? If proline is so disruptive, well, what's it doing inside of this helical structure? Well, it turns out it's everywhere in this helical structure. Okay? And what's interesting about it is wherever we see a proline, we almost always see the glycine. Pro, gly, pro. This is a little bit of a clue as to what's going on. Proline being very inflexible, glycine being very flexible. Put the two together, helical structure becomes possible. Very, very possible. Okay? So this is one of the reasons it's an odd fibrous protein. Now, proline, as I said, gets modified. It gets hydroxyl groups put onto it, as does lysine. And it turns out that those hydroxyl groups are reactive towards each other. Hydroxyl of a proline on this strand over here, or this bundle over here, may react with a hydroxyl on a proline on this bundle over here, and when they do that, they split out water and make a covalent bond. Covalent bonds, we remember, are very, very strong. Why is that important? Well, I'll tell you why it's important. I see several people in here with long hair. Okay? If you braid your hair, or you've braided somebody's hair, you know that you can take and you can make beautiful braids with the hair, but if you don't tie it off at the bottom with a rubber band, what happens? It will all come out, right? Okay? Well, the equivalent of the rubber band are these covalent bonds that form between these hydroxyl groups in the individual bundles or strands that I'm describing for collagen. What these bonds give to collagen is great strength. And when we talk about holding our body together as a glue, we want great strength. We don't want to fall apart. Okay? Now, I tell you that because vitamin C, I told you, was necessary to make those hydroxyl groups. If we don't have vitamin C, if we are deficient in vitamin C, we develop a condition known as scurvy. And scurvy is not something you want to experience, because what happens is you literally fall apart. Your collagen is very weak. The rubber band at the end of your hair or at the end of your collagen isn't holding stuff together. You're in bad shape. Scurvy was discovered first among the pirates who went off on long voyages with no fruits and vegetables, etc., etc. Those silly pirates. And they went out as big, hunking, ugly, mean old pirates, and they came back a bunch of wimps. They fell apart. Okay? Vitamin C is important for that reaction. It's important for making a strong collagen. Okay. Questions about that? Yes? So vitamin C strengthens collagen because vitamin C is necessary for adding the hydroxyl groups to the lysine and proline residues, and those hydroxyl groups react between the individual strands to help hold them together and make them stronger. Just like if we made a braid, like for a rope, the braid is going to be stronger than individual strands will be by themselves. Make sense? I'm sorry? It's not a catalyst, no. It's a necessary substrate for the reaction. Okay, good questions. All right. So that's what I want to say about secondary structure. Let's turn our attention to tertiary structure. Now, as I said earlier, the higher the level of structure, the further apart the interactions are. All right? This is a structure of a protein that we're going to talk a fair amount about next week. It's called myoglobin. Myoglobin is a protein that is not fibrous. And if a protein is not fibrous, we refer to it as globular. Most proteins in nature are globular. Fibrous proteins are not very common in terms of number. They're common in terms of abundance, like we have a lot of collagen, but collagen is only one protein, right? Most proteins are globular. Well, what does it mean to say something is globular? Well, I'd like to just be silly and say, if we were to actually look at this protein sitting on the stage right here, it would look like a glob. Okay? It would not look very regular in structure. It wouldn't have nice, pretty coils going forever and ever and ever. But it would have little coils that would go for a ways, and then we would get a bend. And now you know how bends happen. Mercedes bends, ha-ha. Bad joke, okay? You guys want a joke? You ready for a joke? Okay. I tell this one to my class all the time. I love this joke. It's my favorite joke in the world, all right? If you ever watch my videos, you probably have heard this joke, so if you don't give away my punchline. This is about the hitman known as Artie. How many people have ever heard the joke? A couple. Oh, okay, not too many. All right. So there's this little guy, he's a hitman, right? His name's Artie, and he decides he wants to make it big in the hitman business. So if you want to be big in anything these days, what you have to do is you have to advertise. So he takes a little business card, and he puts on there, we'll kill somebody for cheap, right? And he posts it all over his neighborhood, right? And so he's sitting at home one night, and he gets this phone call. And somebody's gotten his business card, and they call up and say, hello, are you Artie? And he goes, yes, yes. And he said, well, I've got somebody I'd like you to kill. He says, okay. And he says, but how much do you charge? He said, well, I'm just getting started. And he said, I'll do it for $1. This guy's, okay, yeah. So Artie says, who do you want me to kill? And he says, well, I want you to kill my wife. Okay? It's always the wife, right? It could be the wife wants to kill the husband. Doesn't matter, okay? I want you to kill my spouse, all right? I want you to kill my spouse. All right, well, I can do that. Well, is any way you'd like me to kill them or anything? And he says, yeah. He said, I'd like you to strangle them. I said, okay. He writes this all down. He said, and where might I find them? Well, my spouse is down at the grocery store right now. Could you go down and do that? Artie says, sure, why not? So Artie goes trucking down to the grocery store, and he looks, and he sees in the grocery store, he sees this person that meets the description. My career is starting right here, right? Right here in the produce section, right? So he goes up and he grabs the spouse and he starts strangling and kills the spouse right there in the grocery store. Yes, he's done it, right? Oh, no. Somebody saw him, right? Somebody saw him. So he's got to do something. So he goes over and what else is left to do? You can't have any witnesses. So he goes over and he grabs this person and he strangles them. Whew, that was close. Oh, no. Over here, right? My lucky day, right? And I start on my career as a hitman, and I get a witness. So he goes over and he grabs this other person and strangles them right there. And he looks around, and there's nobody. Yes. So he goes running out the door, but the police catch him. And the headline in the newspapers the next day says, Artichokes 3 for $1 at the grocery store. Thank you. You see why that's my favorite joke, right? Artichokes 3 for $1 at the grocery. It takes a while to set that one up, but it's a good joke. Don't tell your friends. That way when they take this class next year, they won't have heard the joke. That'll be good. Okay, back to tertiary structure. So, tertiary structure arises because of Mercedes-Benz, right? And the joke still doesn't work, I guess, after that joke. All right, what do we have are alpha helices that go for a ways, and then they turn. Then they go for a ways, and then they turn. They go for a ways, and then they turn. Those turns give rise to a three-dimensional structure. When we think about the three-dimensional structure of a protein, the turns are really critical because slight changes in those turns point the helix in a very different kind of a direction. Okay? Well, what matters is the overall three-dimensional nature, the three-dimensional structure of that protein. And this protein, when it's in its proper three-dimensional form, carries oxygen. That little thing right here is known as a heme group that we'll talk about later that carries oxygen. Okay? So globular proteins have a three-dimensionality to them. They have more than just a fibrous nature to them. And they also have some bias when we talk about what amino acids that they have within them. Notice here, blue is charged, yellow is hydrophobic, and white is other. Blue charged, yellow hydrophobic, and white other. Blue charged, white hydrophobic, yellow hydrophobic, white other. Okay? What you see on the screen for this particular globular protein is that there's a bias in the amino acids in this overall structure. Polar or charged things tend to be on the outside of the protein. Nonpolar, uncharged things tend to be on the inside of the protein. That makes really good sense. Most proteins are dissolved in water in the cell. And for them to dissolve in water, you want to have the polar and charged groups interacting with water. And the things that don't like water will fold unto themselves and avoid the water. There's no water on the inside of this protein because these hydrophobic groups have squeezed it out. They've squeezed it out. Okay? Not all proteins are dissolved in water. We'll see some exceptions later. And when we see those exceptions, we'll see that they're reflected also in the location of amino acids within them. Okay. In fact, I said later, here's one right here. Here's one called porin. Porin is a protein that is found in membranes of cells. Membranes of cells are fairly nonpolar. So we see on the outside of this guy, the part that's interacting with the membrane, it's fairly nonpolar. On the inside, porin, what it does is it allows water to pass through it. It's a pore. All right? And we see on the inside of this guy, here are the hydrophilics, or the polar charge groups that allow water to interact and come in through there. These last two figures I've shown you are probably the best examples I can give you of structure and function. Okay? Now, I did bring up a song, so I think we'll finish today with a song, and then we will talk more about this next time. Another easy song to sing is called "O Little Protein Molecule." O little protein molecule, you're lovely and serene, with 20 Zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats, arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. And tertiary structure gives polypeptide zing because of magic that occurs in protein folding. A folded enzyme's active and starts to catalyze when activators bind into its allosteric sites. Some other mechanisms control the enzyme rates by regulating synthesis and placement of phosphates. And all the regulation that's found inside of cells reminds the students learning it of pathways straight from hell. So here's how to remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. We'll talk more about those later. So